In this video tutorial, we will show you the steps necessary to clean up your samples after hybridization using the prep station. To process your samples on the prep station, you will need to have your hybridization reactions and you'll need to have a master kit. The master kit contains all the materials necessary for processing your samples. We supply the master kit in 3 different temperature boxes. At room temperature, there is a box containing tips, tubes, hybridization buffer, sheaths, and other materials which are stable at room temperature. The next box contains 96 well plates, all of the reagents, magnetic beads, and other buffers necessary for cleaning up and electrostretching your samples on the cartridge. The third box is stored at -20 C. This box contains 4 individually wrapped cartridges. This shows what the deck layout of the prep station looks like. There's a place for the tip waste and for the liquid waste at the front center of the deck. There's a place for putting empty strip tubes and your samples. The reagent plates go on the right side of the deck. There's a place for the tips and foil piercers, a place for the tip sheaths, which are temporary holding areas for the pipette tips, and there's a place for putting the cartridge and where the electrode closes down over the cartridge for the electrostretching steps. The prep station is controlled using a touch screen, which will walk you through all the steps necessary for a successful run. When you're ready to start processing on the prep station, you simply push the Start Processing button. The next screen you see will remind you to remove the reagent plates and the cartridges from their respective storing temperatures and let them come to room temperature before starting. The reagent plates are supplied with a plastic lid. Take off the plastic lid, but do not remove the foil seal on the top of the reagent plate. The plate should be spun before use for best results. Spin them at about 2000 RPM or 2000 G for a minute or two, and make sure that the magnetic beads are all at the bottom of the plate. The cartridge is supplied in a sealed bag. Keep the cartridge in the bag until it comes to room temperature. This prevents premature hydration of the streptavidin layer and makes sure that all the binding can happen uniformly on the cartridge. Pushing the next button takes you to the next screen. Follow the cartoon on the deck and the blue highlights to show where the next set of materials needs to go. When you're comfortable setting up the prep station, you can bypass these screens by hitting the Finish button. This highlights only the most important screens and allows you to set up your prep station run much faster. The prep station will prompt you to make sure that the liquid waste and tip waste has been cleared before starting a run. Remove your samples from the heat block and spin them briefly to collect condensation and put them on the deck. Take the lids off of your samples before putting them on the deck. Select the number of samples you would like to process. If you are processing all 12 lanes, simply push the Select All button. Place the reagent plates on the deck. The sticker with the lot number should be facing you. A metal pin in the upper right-hand corner of each plate position will make sure that the plate fits correctly on there and that it is in proper orientation for your sample processing. Put the rack of tips and foil piercers on the deck. Note that the gray foil piercers should be facing the front of the deck, facing you. Place the tip sheaths, which are temporary holding areas for pipette tips, in the center of the deck. Place 2 empty tube strips in the heat block in the upper left-hand corner of the deck. Close the metal plate over the heat block and your sample tubes. The prep station will not run if this metal plate is not down over your samples. Place a cartridge in position. The prep station will not run if a cartridge is not in place. When putting the cartridge on the deck, be very careful that you don't accidentally bend the wires of the electrodes. There will be a slight clicking sound when the cartridge is in place and the switch is activated. Close the electrode fixture down over the cartridge. Again, be very careful when you close this, and if you encounter any resistance, stop, reposition the cartridge, and try again. Next, enter an email address for notifications when a run is complete or if there is an error. If this email is not set up, please let NanoString know, and we will contact your IT department and main users in your group in order to set this up correctly. Finally, close the door. The prep station will not run if the door is open. Then push the Start button. The prep station will calculate the length of time necessary to process the run and initiate a series of checks to make sure that the prep station is operational before doing anything with your samples. If it encounters a problem, the prep station will stop, send an email and/or audible alarm, allowing you to intervene and correct the situation. When the prep station is finished, it will send an email, an audible alarm, and will have a blue screen showing the length of time since the run has completed. At this point, you can take the cartridge out and cover it with the tape sealant that we provide for you. At this point, it is a good idea to label the cartridge with a date and a title for this particular cartridge. The cartridge can be stored at 4 degrees until it is ready to be scanned on the scanner. Do not let the cartridge sit for more than an hour on the deck of the prep station without covering it with the tape sealant. Leaving it uncovered will result in evaporation and prevent you from collecting data from that cartridge.